Thank you so much. Okay, so why would these reactions not take place without an enzyme? And the answer is because these well, remember first off that the chemical reactions we're talking about are either going to be building a bond or breaking a bond. So that's why we're seeing here, we're looking at a bond between two molecules and we want to break that bond. So obviously, biochemically, it wouldn't make a lot of sense for that bond to just break spontaneously, right? We store energy in chemical bonds. That bond's going to stay put until we want it to break. And so the strength in that bond, basically the amount of energy it would take to break that bond, is what we call the activation energy. So every molecule and every bond has a certain amount of activation energy. And this activation energy, for the most part, is so high that this reaction, breaking that bond or building that bond, won't happen unless we facilitate it, right? So again, this bond is not going to be broken because it's so strong. It has a high activation energy. We have to actually have something that can allow us to overcome that activation energy or provide the energy necessary to break that bond. So that's the purpose of an enzyme. So when we say enzymes are catalysts, they basically speed up this chemical reaction, but really what they're doing is they're allowing this chemical reaction to take place, because what you can see that this enzyme did is it lowered the activation energy of this reaction. So this can happen in a lot of ways. So for example, if you're talking about breaking a bond, what an enzyme can do is kind of like move the molecule so that bond is no longer like chemically stable. And the two compounds that are forming that bond are just not really easily able to connect to each other. So again, destabilizing that bond would allow that bond to break. It would provide enough energy to overcome that barrier of activation energy. So again, enzymes are going to speed up reactions. That's why they are called catalysts. And again, a catalyst is something that speeds something up. In this case, we're talking about chemical reactions. But in all reality, right, so you're looking at this reaction of breaking that bond. In all reality, that's probably not going to happen without the help of an enzyme. I also want to mention that it is a common misconception that enzymes only serve to break bonds, but we also need enzymes to help build bonds. So you have enzymes that are very important for catabolic reactions, so such as the one we're looking at in this picture, but you also have enzymes that are really important for anabolic reactions. And again, these are always named for what they do. We've already talked about several. So for example, RNA polymerase is an anabolic enzyme because RNA polymerase takes nucleotides and builds bonds between them to make RNA. So that, remember we talked about the fact that if you hear the term polymerase, ACE tells us an enzyme, polymer tells us we're building a large molecule, and then RNA tells us what that large molecule is. So RNA polymerase is an enzyme that facilitates bonds being formed between nucleotides in RNA. DNA polymerase facilitates those bonds between nucleotides to create DNA. So we have enzymes, again, for both anabolic and catabolic reactions. Enzymes are arguably one of the most important biological molecules in a biological system. We can't live without them. We can't live without them. Okay. So, as we mentioned, all of our enzymes have the structure where they have the enzyme and then they have a little pocket called the active site. And that active site is where our substrate fits in. So I just want to remind you, if we're like building or breaking a bond, a substrate can be, it's not always just like one compound. So you can see that this entire thing is the substrate for this enzyme. So it can be pretty large molecules. But the substrate, which is the reactant of the reaction, fits into the active site of an enzyme. So that would be the normal binding of the substrate. If this took place, the reaction would occur. A lot of our enzymes rely on more than just the substrate binding to the active site for the reaction to occur. So if you haven't noticed, whenever we talked about transcriptional control and translation and control, all that stuff, our body and cells in general are very, very, very good at kind of regulating things. So not only does like substrate binding to the active site, that's not necessarily enough for us to get this reaction to proceed. Usually, just like with transcription, we have to say like, yes, go ahead, or no, you don't do this reaction. Because enzymes are so important, usually we have a lot of them present in a cell. So because we have a lot of them present in a cell, we want to be able to turn them on and off when they need them without necessarily regulating them when they're transcribed. So we're going to talk about a bunch of ways that can happen, but the most common way is by directly controlling the enzyme. So you can do this via the process of activation. So with activation, you would have your substrate bind your active site, but then you would have another molecule that basically just serves to just like turn the enzyme on. There's a lot of different molecules that can do that. We'll talk about some on the next couple of slides. It's like coenzymes and cofactors, prosthetic groups. Phosphorylation is also an example of a cofactor that basically serves to turn on enzymes. But it turns out that the most common way that enzymes are regulated is basically just by like turning them off. And this way we can turn them on whenever we want. So it's kind of the idea that the body has, our cells in general have the ability to basically just keep everything off and then turn something on when needed. And that's the case with enzymes as well. So enzymes are turned off via the use of what are called inhibitors. So inhibitors are just small molecules that bind to an enzyme. And we can have two different types of inhibitors, and they're basically classified by where on an enzyme they bind. So we have what are called competitive inhibitors. So you can see in the picture on the left, competitive inhibitors bind to the active site of an enzyme. So if you have a molecule binding to the active site of an enzyme, that means its substrate is not going to be able to bind to the active site. So competitive inhibitors literally like physically block the ability of the substrate to bind to the active site. So that will prevent, if you're preventing the substrate from binding to the active site, that means if the substrate can't bind to the active site, the reaction will not take place. So competitive inhibitors physically block binding of the substrate to the active site. They're called competitive inhibitors because they're like competing for that active site. And then you also have what are called non-competitive inhibitors. And non-competitive inhibitors, they're called non-competitive because they don't compete with the substrate for the active sites. That's because they actually don't bind to the active site. They bind to a site that is away from the active site. And this site is called the allosteric site. So what this does, allosteric, that terminology, actually means that it does still affect the shape of an enzyme. So as you know, throughout all of biology, structure equals function. So if something binding to an enzyme, like a non-competitive inhibitor, affects the shape of an enzyme, it will affect the function of that enzyme. So you can see kind of what happened here is even though the non-competitive inhibitor did not bind to the active site, its binding did change the shape of that active site. So you can see here initially the enzyme had like the active site was kind of open, but what happened here is this binding of the non-competitive inhibitor sort of formed like a little indentation, basically closing the active site. And so what non-competitive inhibitors do is they don't physically block the active site themselves, but they may change the active site, basically closing the active site so that the substrate can't bind. But again, no matter what, if you're preventing the substrate from binding to the active site, you're going to make it to where that reaction can't take place. Okay, so we can control enzymes by turning them on via activation or turning them off via inhibition. Competitive inhibitors bind to the active site. Non-competitive inhibitors bind to the allosteric site, but change the shape of the active site so the substrate can't bind. All right. We do have other ways that we can kind of impact. Oh, I had a question. Does this also happen with the competitive inhibitors? Good question. No. So you can see here, competitive inhibitors bind to the active site, and they just function to kind of physically block the binding of a substrate. So that's kind of how they're classified. Allosteric inhibitors kind of, as a group, change the shape of the active site, whereas competitive inhibitors are classified as just blocking the active site. At least that's what I understand. But I'll have to see if any competitive inhibitors change the shape of the active site. I guess what I'll say is I don't think they do change the shape of the active site, but they just functionally block it. Good question. Oh, sorry. The question was the function of enzyme change with competitive inhibitors. Oh, great question. No. The enzyme's function is always very specific to its substrate and whether it's going to build or break a bond. So this enzyme is going to act on this substrate either to build or break a bond with this somehow with the substrate. When you have a competitive inhibitor bound to the active site, the substrate cannot bind. So the reaction that was going to take place on the substrate just cannot function. So it's not that the function changes, it's just that that enzyme literally just can't do its job. It's not going to be able to perform the reaction. So it's not that the reaction that it performs changes. The function is not changed. It's literally just, is it on or is it off? I hope that makes sense. It's a good question. But it will not change its function. All right. There are certain things that can change enzyme function, but we're not going into that today. Well, I guess we kind of can because we can talk about impacts on enzyme function. So typically when we talk about impacts on enzyme function, what we're typically talking about is how can we get this enzyme to perform better, like more quickly or more efficiently or something like that. We have what are called kind of like enhancers of enzyme function. So you can have a couple different types of these chemical enhancers. You can have what are called prosthetic groups, which are small molecules that like permanently attach to an enzyme, allowing it to function. So your question was, does the function of an enzyme change with competitive inhibitors? And the answer is competitive inhibitors would not change the function of an enzyme. That enzyme would it would just turn that enzyme off. But what prosthetic groups can do is they can modify an enzyme to where they could potentially allow an enzyme to go from performing one job to performing a different job, slightly different job. So competitive inhibitors don't necessarily change the function of an enzyme, but prosthetic groups can allow us to use enzymes for more than one reaction. Usually though, what prosthetic groups do is they kind of enhance the function of an enzyme. We also can have cofactors. This is molecules like iron or zinc. A lot of times these are metals. Magnesium is another example. And these bind temporarily to an enzyme, allowing it to function better. There are several enzymes that will not function if you don't have these ions present. And in fact, there are several bacteria whose enzymes won't function if they don't have certain inorganic ions present. So the organism that causes the plague, which is called Yersinia pestis, actually, in order to infect an organism, has to use a very specific enzyme, but that enzyme doesn't function without iron. So the plague interacts very closely with blood, and so there tends to be a lot of iron on red blood cells, obviously with hemoglobin. But what we can do is in a lab, if we want to impact its ability to infect cells, we can actually restrict the amount of iron present. And if you restrict the amount of iron present, this means that it doesn't infect as readily. It's pretty interesting. So that's just an example of a bacteria not functioning well in the absence of a metal like iron. But several of our enzymes don't function very well as well. I mean, again, with hemoglobin, hemoglobin's a protein, but it needs iron. And then we also have what are called coenzymes. Cofactors are inorganic ions. Coenzymes are organic molecules. So phosphate, like I talked about with activation, phosphorylation, actually that's a cofactor. But coenzymes would be like an organic molecule. So these are going to be like vitamins. Several vitamins function in this way. The one that was given as an example the other day, where is it? I just had a really good question. Where'd my notes go? Here we are. Somebody had a really good question the other day about ubiquinol. So ubiquinol is not the same as ubiquitin ligase, but what ubiquinol is, we will learn specifically through this lecture what it does, but ubiquinol is actually a vitamin, and so it serves as a coenzyme. It allows a certain reaction to take place. So we'll learn through this reaction what reaction that allows to take place. All right, so it's an organic thing. But all of these, whatever kind of thing we're talking about with enzyme function, all of these are chemical ways. So whether it be just like a small molecule, an inorganic molecule, or an organic molecule, all of these somehow like affect an enzyme's ability to function, typically speeding it up or increasing its efficiency. And just like a lot of reactions won't take place without an enzyme, a lot of enzymes won't be able to function without their coenzyme or cofactor. So they are extremely important. As well as that, there can be physical factors that impact enzyme function. So for example, heat. We know that heat increases enzyme rate. So you can see that here, but this is only to a certain extent. So this is looking at, on this axis, we have the rate of chemical reaction taking place. And on this axis, we have temperature over time, or this is over time, yeah, temperature. And you can see that the rate of a chemical reaction, how quickly it proceeds, increases to a certain temperature. So as temperature increases, the rate of the chemical reaction, so how quickly it's taking place, also increases. But note, right around here, 37 degrees Celsius, enzyme function, once you kind of like get too hot, enzyme rate starts to decrease. And one of the reasons we see that is because enzymes are proteins, and proteins can denature outside of a homeostatic range. So to a certain extent, heat can increase the rate of reaction, but once you kind of cross that threshold, enzymes start to not function because they start to denature. So again, this kind of, we talked about with homeostasis, we talked about fever. It is believed that one of the reasons a fever can help with sickness is because if you have your fever increase, that increases the temperature of your body. So that would allow your enzymes, such as enzymes that help with immunity or fight infection, it allows them to kind of work more quickly and more efficiently to kind of destroy the infection. However, you talk about that 104 being the threshold. 104, if you go above that, your enzymes are not necessarily functioning better. They start to denature, so they don't function at all. So heat can help increase enzyme function to a threshold. The same with pH. So there are actually certain enzymes that will not function outside of their homeostatic range of pH. For example, the enzymes in your blood function at a pH of 7, but the enzymes in your stomach actually require that acidic pH to function, like proteases that destroy proteins, etc. All right, so we are going to be moving forward talking not only about single enzymatic reactions, so converting this to this in one reaction. In fact, we're going to be talking about what are called metabolic pathways, which are where we have a lot of chemical reactions paired together. So this reaction takes place, and then this reaction takes place, and then this reaction takes place. So when that happens, when we have reactions that are coupled, we have to ask, so how can they be coupled and how can we regulate this? So metabolic pathways can be regulated in a lot of ways. So they can be regulated genetically, we can regulate the individual enzymatic reaction, but at least one way that we can link them is basically by having the products of one reaction be the reactants of a second reaction. So that's what you're looking at down here. In this picture, you have an example of a metabolic pathway. So for example, like if you've studied cellular respiration before, cellular respiration is typically divided into 3 or 4 steps, but those 3 or 4 steps all happen sequentially. That is what makes it a metabolic pathway, rather than just like one metabolic reaction, is that they are linked. And they are linked because, as we will study soon, we have reactants that go into the reaction, for example, like let's say a sugar, glucose, and the first reaction takes place and generates a product. So in the case of like cellular respiration, we go through glycolysis, we turn glucose into pyruvate. But then the product of that reaction is actually the reactant for our second reaction. So for example, the intermediate stage, or if you've studied it with just 3 reactions, like the Krebs cycle. So pyruvate would go into that second reaction as a reactant, and then we would get a product. In this case, electrons, which go through the electron transport chain. So with metabolic pathways, that's when we have reactants of one reaction produce products. And then those products of the first reaction are the reactants for a second reaction. So that's how we make sure that they are linked. They are linked because the products of the first reaction become the reactants for the second reaction. And again, that all has to do with enzyme specificity. This enzyme is specific for this substrate. This enzyme is specific for this substrate. So we can couple those. One way that we can regulate that by saying, how do we know we have enough product, or how do we know this pathway has taken place basically enough, is via the process of feedback inhibition. So feedback inhibition, what would happen is we would have this reaction take place, this reaction take place, this reaction take place. We would get our final product. And let's say we have like enough of our final product, that what can happen is that final product can serve as an inhibitor So either competitive or non-competitive, but remember, just inhibitor means it stops an enzyme's function. This product can serve as an inhibitor for one of our previous enzymes. So this first enzyme. And if this inhibits this enzyme, then this reaction will not take place. So that's a way for us to say, like, for example, if I'm consuming a lot of sugar, when you consume a lot of sugar, you can do two things with it. You can use it or you can store it. So your body has to decide, do I need to use the sugar right now or do I need to store it? So if you use sugar, right, if you are needing a lot of energy, say you're going to run a marathon, you need that sugar turned into ATP to fuel your body. So you need cellular respiration, a catabolic reaction to take place. But let's say that you consume a lot of sugar and you're just going to be sitting around watching Netflix all day. You don't necessarily need that energy now, but what you would need is basically your body says, okay, well, I'm just going to store it for later. And it stores it by combining glucose together to form a molecule stored in your liver called glycogen. So what can happen is your body gets sugar, let's say sugar is A, and you go through the steps of cellular respiration, your product is ATP. Let's say your body's like, listen, I have all the ATP I need to function right now. We're not going to run a marathon. We don't need any more energy. Let's save it. So what can happen is a product, whether that's ATP or pyruvate or whatever, can actually stop this initial reaction from taking place. It can like inhibit the first reaction, cellular respiration, and that would prevent glucose from being broken down, and then instead glucose would hang out and be stored. So that's just a way that we can say, okay, we have enough product, we've reached a threshold, let's like stop breaking it down. Right? So again, that's called feedback inhibition. It's when a metabolic pathway, one of the kind of like terminal products, serves as an inhibitor for one of the earlier enzymes. All right. So I do want to just mention that metabolism in general is the sum, so just again, we've already talked about this, but I think it's important to reiterate it constantly, apparently. Metabolism in general is the sum of all of the chemical reactions in a cell or an organism, so how we are transforming that energy. And we have two general ways to do that. We can take large molecules and break them down into smaller molecules via catabolism. Remember that catabolic reactions, because we're breaking chemical bonds, releases energy from those chemical bonds. But the product of catabolic reactions, being small molecules and energy, are actually the reactants for anabolic reactions, where we use small molecules and use energy to build larger molecules. So catabolic and anabolic reactions are intimately linked together. They are forming these really large metabolic pathways where we're breaking molecules down and using those products to build other molecules. Catabolic reactions serve to release energy, but then we can use that energy to build other molecules. So metabolism is this very intimately linked cycle. So we're going to talk about that later, probably Thursday, when we talk about cellular respiration, because cellular respiration is noted for its ability to produce just like a ton of ATP, which is its primary purpose, right? To break sugars down into ATP. It is a catabolic process overall for that reason. But it is also a central pathway in a cell or organism, because as it breaks stuff down, it actually releases a lot of small molecules that are extremely important for anabolic reactions. Without cellular respiration, we actually wouldn't have a lot of the stuff we need for creating proteins or creating sugars, actually, and creating amino acids. So we need these catabolic reactions for these anabolic reactions. We need anabolic reactions for catabolic reactions. So, you know, cue the Lion King. It's a circle of life. All right. Okay, so we're going to switch gears here a little bit. So we've talked about all the type of chemical reactions, how there's mediated by enzymes. But honestly, like, we keep talking about energy without ever fully kind of discussing like what the heck we're talking about when we're talking about energy. Like, what are we talking about? Well, there's a lot of different types of energy that we can classify in a biological system. One of the most common types of energy that we are talking about in a biological system is the energy that is stored in a chemical bond. So we'll talk about the other type of energy that we talk about in biological systems here in a minute. But for the most part, we really talk a lot about the energy that is stored in chemical bonds. So just a reminder that if energy is stored in chemical bonds, it's stored, so that's potential energy. And when bonds are broken, we release that energy, and that is usable energy, which is kinetic energy. So what are we like actually talking about storing and releasing from chemical bonds? Well, if you have taken chemistry, are currently in chemistry, or will have chemistry, what you should know is that all chemical bonds involve the interaction of two molecules, but their interaction really depends upon their subatomic particles. Electrons are extremely notable here. And so in the case of chemical bonds, because electrons are so important in the formation of chemical bonds, what we really are kind of talking about when we talk about biological energy is electrons. Electrons have a lot of energy, right? That's where our cellular source of energy is. So when we have stored energy in a chemical bond, what we're storing is electrons. And when we break a chemical bond, what is released is electrons. So why these are so energetic is because individually they have a lot of chemical energy, and we can use them in the formation of other chemical bonds, or actually to facilitate breaking other chemical bonds. So individually, electrons have a lot of power. So that's really a lot of times what we are discussing when we are discussing energy stored in chemical bonds or released from the chemical bonds. We're talking about electrons. Okay, so again, electrons are stored in chemical bonds. That's potential energy. When it's released, that's kinetic energy, and that's the release of electrons, which we're going to use in a different way. So we'll talk a lot about moving forward how we use those electrons. So that's one type of energy we're going to talk about, is energy that is stored in bonds in the form of electrons. Another type of energy that we're going to talk about is what you're probably used to hearing referred to as cellular energy, and that is energy in the form of ATP. So ATP stands for adenosine triphosphate. An adenosine triphosphate is like two sugar molecules, adenosine, which contains a nitrogen, and a ribose, and that tri here is referring to tri meaning three, and what that's referring to is that we have three phosphate groups attached to that ribose sugar. So the reason ATP is kind of like our gold standard of cellular energy has a lot to do with the fact of basically how these phosphate groups interact with each other and form these bonds. What basically happens is that when we have these sugars and we have these phosphates, the interaction between all of these molecules kind of pulls the electrons toward the center of the molecule due to electronegativity. But what that means is that this bond between the second and third phosphate in this molecule, as you can see in the picture, has a lot of energy. And again, we're talking about energy in a bond. So we have energy, electrons stored in that bond, but when that bond breaks, it's going to release electrons that can be used either to build or break other chemical reactions, or basically kind of be used in chemical reactions. So we have a high energy bond. What we mean by high energy is that it is very willing to kind of break and have other molecules use the electrons stored in that bond. So it is a very great source of cellular energy because it's kind of willing to donate that energy, those electrons, to any other reaction very easily, right? So that bond is basically relatively unstable. It's willing to break and help out other reactions whenever they need it. So that's ATP. So ATP is also really nice because it's a relatively simple molecule. Again, 2 of these molecules and then 3 phosphates. It's the interaction between the second and the third phosphate where we store all of our energy. So basically, if we have triphosphate, that's like a charged battery. It can contribute that electrons to whatever reaction needs it. But if you break that bond, you can't see me put my, I tried to put my hand up to the screen, it was so dorky. But if you break that bond and release that third phosphate, right, that's kind of like an uncharged battery. There's still a little energy in this bond, but not really enough to contribute to other biological reactions. So you have 2 phosphates, that would be adenosine diphosphate, but all we need to do to like charge that battery up is add a phosphate and make it ATP. So we consider ATP as sort of a rechargeable battery, and we can kind of recharge it. We recharge it by using energy from other reactions. So we're actually going to move forward soon, talking about the different ways that we can generate ATP. But basically, we just take energy from some source. So this says from cellular respiration, although that's just one source of energy. We can have energy from any source. This could also be maybe from the sun, right? Wherever you're getting energy. And what you do is you use energy to build a bond between adenosine diphosphate and another phosphate. This is a chemical reaction. We're building a bond, and so we do need an enzyme. The enzyme that does this is an enzyme called ATP synthase. Synth is kind of the prefix meaning synthesis. ASE, the suffix tells us that it's an enzyme. So it's an enzyme that synthesizes something. Specifically, it synthesizes or creates ATP. So we are using energy from some source in order to build a bond between adenosine diphosphate and another phosphate to create ATP. So then that ATP, again, is a really great source of cellular energy for whatever else we want to use it. All we have to do to use that energy is break that bond between that second and third phosphate, and that will release that energy for cellular processes. But if you release that energy, then if you break that bond, then you're left with ADP and phosphate, which we can use ATP synthase and a little energy to regenerate that bond. So ADP, because it's not really used for anything else except for cellular energy, really function as sort of this sort of like rechargeable battery that's sort of just like hanging out in the cell. We can use it and recharge it and use it and recharge it kind of as many times as we need. So that's why we love ATP. It's really, really, really efficient because it's got this high energy bond, but also it's a rechargeable battery, right? We don't have to create it constantly. So our question should be focused here, which this doesn't give a lot away, but our question should be, okay, great. ATP is important, I got it. Where do we get the energy to create that bond? And the answer is we can get energy to create ATP via 3 primary ways. And these are all, again, generating ATP. So remember that we take ADP and add a phosphate. So that addition of the phosphate is called phosphorylation. Anytime you add a phosphate to something, that's called phosphorylation. So in this case, because we're making ATP, we're adding a phosphate, so we're phosphorylating ADP. So that's kind of gives you the terminology here. You'll notice that every single one of these is called some sort of phosphorylation. That's because every single one of these pathways is going to be adding a phosphate to ADP to create ATP. So the big thing that differentiates these pathways is where are we getting the energy to form that bond. So you'll notice that it tells you exactly where that energy is coming from. Number 1 says it's substrate level. So think about what a substrate is. It's what is acted upon by an enzyme. It's also the reactant of a reaction. So this tells you that this phosphorylation, the energy is going to be coming directly from the reactant of a reaction, okay? So that's substrate level phosphorylation. Oxidative phosphorylation tells you that the energy for phosphorylation of ADP to create ATP comes from oxidative reactions. So we're going to talk about redox reactions there. And then this one, photophosphorylation. Photo means light. So in this case, we're talking about energy to generate that phosphorylation comes from the sun. Okay, so I actually need to use the restroom really fast. So I am going to pause this and be right back. I will be right back. Okay, thank you. Sorry, I had to go. Okay, that's what we get from live lectures. Just imagine if we were in class, it'd be like running out of class. All right, so let's get back to it. Okay, so we were saying that we have 3 ways of making ATP. We have substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. All of these are serving to create that bond and generate ATP, so add that phosphate group. That's what we refer to with phosphorylation. So the big thing that differentiates these 3 different mechanisms is where that energy is coming from. Okay, so let's go through these. So the first type of phosphorylation we can utilize is called substrate level phosphorylation. So this is called substrate level phosphorylation because basically the energy that we need to generate the bond between ADP and a phosphate is released directly from the breaking of a bond in a substrate for a reaction. So that's what we're looking at here. On the bottom of the slide, you can see a substrate for a reaction. So our reaction is basically taking this substrate or this reactant, and we're going to break this bond, and we'll have two products. But remember, we talked about that energy is in the form of electrons. So and that energy is like usable energy. So what happens with substrate level phosphorylation is we have a bond broken, electrons are released, and those electrons are sufficient energy to add a phosphate to ADP. Easy peasy, all right? And sometimes substrate level phosphorylation, we can also get a phosphate released. So if there's a phosphate associated with this substrate or this reactant, you might also get not only electrons released to kind of provide the energy for the bond, you might also get the actual phosphate released, which is pretty common as well. So here's the big thing with substrate level phosphorylation. We just saw that when a bond was broken, that gave us the energy to form ATP. It happened right there. So this is called substrate level phosphorylation, or SLP, because this phosphorylation is happening at the level of the substrate, right where the reaction is taking place. So we're going to differentiate this from the other two mechanisms, that oxidative phosphorylation and photophosphorylation, because neither of those have that phosphorylation event just like right there. Okay, we'll still see chemical reactions, but both oxidative phosphorylation and photophosphorylation require basically like two steps. Okay, substrate level phosphorylation requires one step, breaking a bond. And that breaking of a bond releases enough energy that we can fuel the phosphorylation of ATP. All right, so that's substrate level phosphorylation. That was our first way that we can generate ATP. We break a bond, releases enough energy to form the bond in ATP. The next way that we can generate ATP is via a process called oxidative phosphorylation. So oxidative phosphorylation kind of tells you, again, we're still phosphorylating ADP to create ATP. But this is telling you where the energy is coming from. And again, we're talking about energy in the form of electrons. Our electrons, in this case, are coming from oxidative reactions. So this is where we get into a little chemistry, but hopefully we can discuss enough to where even if you haven't had chemistry yet, it's not a big deal. So one really, 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 really important chemical reaction in biological systems are paired reactions, and these are called redox reactions. So I'll go into these more specifically on the next slide. But redox reactions are paired reactions when we have two reactions that depend on one another. And redox reactions are basically reactions that involve the loss, so releasing, and then the subsequent catching or gaining of an electron. So in this case, you'll have like one molecule release an electron and another one kind of grab that electron up. So anytime a molecule releases an electron, that's called oxidation. So a molecule that is oxidized means that it has given up an electron. But then electrons can do a lot of damage if they're kind of just like freely hanging out. So if a molecule is oxidized and releases an electron, you're going to have another molecule that serves to kind of like grab up grab that electron. All right, bind it up. And if another molecule grabs an electron or gains an electron, because electrons are negatively charged, if something gains an electron, its charge is reduced. So when we have a molecule release an electron, it's oxidized, but when another molecule grabs that electron, it's reduced because its charge is reduced. So with redox reactions, you have oxidation, so electrons are released from a molecule, and then you have another molecule reduced. So this is really important because oxidative phosphorylation, right? We've talked about substrate level phosphorylation, and in that case, what happens is something is oxidized, an electron is released from a bond, but then that electron is used to create ATP. So it's not a redox reaction. Well, I mean, technically something is oxidized, but that electron directly contributes to the formation of ATP. In the case of oxidative phosphorylation, what happens is we have a bond broken. We have electrons released, but we have certain molecules grab those electrons. These are called electron carriers. And these include things like NAD, FAD, and NADPH. And they carry these electrons. So remember I said oxidative phosphorylation has two parts. The first part is releasing that electron. And the second part is carrying that electron to a different part of the cell where that electron will be used to generate an electron transport chain. So oxidative phosphorylation is a type of phosphorylation that involves an electron transport chain. And that electron transport chain generates a proton motive force, which is going to give us a different type of energy. So that's a lot of terms, but we're going to go through it. All right, so let's look at this. So first off, what are redox reactions and how can we remember this? So as I mentioned, redox reactions are when we have one molecule. So let's say that we have this chemical reaction. We have two reactants. We have A and B, and these go through a reaction. We still have those two molecules, but you can see what's happening here is the electron is being moved around. So in this case, A has an electron, and when it goes through this reaction, you can see that A loses that electron. So if A loses an electron, it is said to be oxidized. You can see oxidized, it's lost an electron. Again, we don't really want electrons kind of like hanging out because they can do a lot of damage. So what happened in this reaction is A lost an electron, A was oxidized, but then B actually received that electron, it grabbed up that electron. And in this case, because B grabbed that electron, B was reduced. Its charge was reduced because it gained an electron. So we, again, redox reactions, you can kind of remember this because of the anagrams. The one I was always taught was oil rig, which stands for oxidation is loss, because if a molecule is oxidized, it loses an electron. So oxidation is loss, and then reduction is gain. Talking about if you have a molecule gain an electron, it's been reduced. So oxidation is loss, reduction is gain, referring to electrons. A friend of mine uses the term, or uses the anagram Leo the lion, says GER, saying like loss of electrons is oxidation, whereas gain of electrons is reduction. So I don't really care how you memorize it. Just know that if a molecule loses an electron, it's oxidized. If a molecule gains an electron, it is reduced. So I had a question. Why did they choose the term reduced to describe an electron gaining an electron? I know, it's so confusing. I get it. The reason they say that a molecule is reduced when it gains an electron has to do with the fact that electrons have a negative charge. And so what happens to this molecule when it gains that electron is its charge is reduced. So that's kind of what we're talking about when we talk about reduction. If a molecule is reduced, it gains an electron, which means its overall charge is lowered. I know it's confusing, but think about it in terms of charges, not necessarily subatomic particles. Yeah, good question. So, all right, so oxidation, we're losing electrons, reduction, we're gaining them. And these are very important for redox reactions. So for oxidative phosphorylation, we will always have a molecule oxidized, and we're always going to have another molecule reduced. And that reduced molecule is going to serve to basically carry that electron to the second part of the requirement for oxidative phosphorylation, which is the electron transport chain. Because remember, oxidative phosphorylation occurs in two parts: redox reactions and an electron transport chain. Okay, so with the first part of that, what happens is you will have a molecule. So we just see a molecule with a bond between it. That bond is broken and an electron is released. So far this is very similar to substrate level phosphorylation, but note the big difference here is that when we break this bond and release an electron, that electron is not immediately used to generate ATP. ATP is not being generated at the level of the substrate in oxidative phosphorylation. Instead, what happens is we have a molecule with a bond, we break that bond, so when we break that bond, that molecule was oxidized, it released an electron, and that electron reduces another molecule. So in this case, what the molecule that's going to be gaining that electron is, we're just going to call it X. So X gained an electron. So there's something that's really important here. So I'm actually going to write that. Oops. Here. Did I do it? So X gained that electron through this reaction. So something that's really important, you'll notice that I say that X gained that electron, so X was reduced. You'll notice that I also have a hydrogen here. So typically, you're not going to see a molecule kind of gain a negative charge and be able to hang out, right? We don't have a lot of just like negative charges or molecules carrying electrons around. In the body, we're all about neutralization. But also, as we will learn, hydrogens are really important for the electron transport chain. So what happens is we have this molecule oxidized. It releases an electron. Some molecule is reduced, so it gains that electron. But since this molecule gained an electron, a negative charge, it actually also grabs a complementary positive charge. So hydrogen ions are perfect for this because hydrogen ions are basically just protons. And remember, protons are our positively charged subatomic particles. So basically what can happen is if X is reduced and gains an electron, we can kind of like balance out that negative charge with a hydrogen. So typically what you'll see is when a molecule is reduced, you'll actually see that indicated by the gain of a hydrogen. So you can see this. So for example, we're going to be talking about cellular respiration. If you've talked about it in previous classes, you know that, and if you haven't, that's okay because we're going to talk about it. But you have electron carriers that are reduced. So for example, NAD is a molecule that is reduced in cellular respiration when glucose is oxidized. NAD is the molecule that grabs up the electrons that are released. But you'll see that when NAD actually grabs that electron, it doesn't become NAD minus, it becomes NADH. And that's because in order to complement the fact that it grabbed up an electron, it also grabbed up a hydrogen. So if you see like NAD or FADH2 or NADP gain a hydrogen, you should know that it's also carrying an electron. It's been reduced. Okay. So again, what we're trying to do overall with oxidative phosphorylation is we're trying to break a bond, release electrons, use a molecule to grab those electrons, and then we're also going to grab some hydrogens. So okay, so like the end result of a redox reaction is we have this molecule carrying an electron. Great, we don't have any ATP yet. So how do we use this molecule carrying an electron and a hydrogen to generate ATP? So it's very important to note, again, oxidative phosphorylation occurs in 2 steps, redox reactions, and then an electron transport chain. So what happens next is those molecules, so I think here we go, I like this better. We have a molecule, we break a bond, we release an electron. We have a molecule reduced, which when it's reduced, it also gains a hydrogen. So I'm using the example of NAD. When it gains that electron, it also grabs up a hydrogen, so it becomes NADH. So that NADH, or whatever electron carrier we're using, then goes to a membrane. So in the terms of cellular respiration or oxidative phosphorylation in a non-photosynthetic animal cell, what happens is it goes, well, I guess in photosynthetic cells, any organism that has a mitochondria, is it's going to go to the mitochondrial membrane. The membrane is extremely important because what it does is these membranes have a series of pumps, and these are hydrogen pumps that are fueled by electrons. So what happens is this NADH, which is carrying an electron and carrying a hydrogen, goes to a membrane. It releases that hydrogen and that electron, and that electron serves as energy to pump hydrogens to one side of a membrane. So why does it matter that they're pumped to one side of a membrane? Because if you recall, we can generate what are called concentration gradients. So this is thinking way back to when we talked about like osmosis and everything. I put a picture in here, but I don't see it. Dang it. But I want you to think about, with these concentration gradients, what I want you to think about is I want you to think of like, let's see. Oh my gosh, get out of here. Ignore my shoe shopping. I want you to think about like a dam. So you can think about like the Hoover Dam. So whenever you're thinking about like here, a membrane holding back a lot of molecules, you can kind of equate that to like a dam holding back a lot of water. So obviously there is a ton of potential energy held back behind that dam. If we were to open that dam, that potential energy would come flying through, and that would be kinetic energy. So what we're doing with oxidative phosphorylation is we're releasing a ton of electrons, and we're basically using those electrons to generate a concentration gradient across a membrane. And what we can do is we can use that tremendous amount of energy. Think about an electron transport chain as like just building the Hoover Dam. It's a Hoover Dam holding back hydrogen ions. And what we can do is ATP synthase basically kind of just like opens up and allows that to flow through, which is a tremendous amount of energy, and you'll see at the bottom here, to generate ATP. So just to revisit oxidative phosphorylation, and we will come back to this on Thursday, but oxidative phosphorylation, the first step is to do a redox reaction, release electrons, harness those electrons, but then the second step is that we carry those electrons to a membrane to generate an electron transport chain. Whereas opposed to substrate level phosphorylation, ATP is generated when that bond is broken. Substrate level phosphorylation, there is no electron transport chain. Oxidative phosphorylation, there is an electron transport chain. Okay, we're out of time. I'm so sad. But I love talking to you guys. But I do want to honor our class time. So I think that's where we're going to call it for active lecture today. But I'll hang out if you have any questions and I can answer those questions. Just know that kind of if you want to move ahead with lecture, I have pre-recorded the majority of this lecture. All I have left to do is photosynthesis. So if you want to work ahead, I will be posting those today. Yeah, if you have questions, send me an email or feel free to post in the discussion board, which is actually, I would love it if you did that, because then on Friday I can do what I sort of similarly did for the genetics lecture, which is go through and see like how many questions we have about whatever and make a whole separate supplemental lecture for that. Whatever you want to do, but just make sure that if you have questions, please ask them so that I can answer them. I want to make sure you understand them.